putting us into the transition here. Sorry, what? Oh, okay. Right, there we go. All right, I hope people can still hear me because I'm going to be introducing this first session as it starts. Our first session, topic A, is also called the 2018 Roadmap. This is about state-of-the-art and promising developments in the main challenge areas for brain emulation. So things like, how do we map the structure of a brain? How do we record the response characteristics of its neurons and synapses? How's a working model generated with parameters defined by those data maps? And how is that brought to life in a real implementation of the whole brain emulation, or in a similar, smaller subset, such as a neural prosthesis? Now I'm going to be introducing our distinguished speakers one at a time. Each speaker is then going to give a presentation. And if necessary, I'll give a brief signal to the speaker when they have about 5 minutes left. And then I'll moderate the Q&A discussion for that talk. The Q&A is going to be an interesting challenge because we'll have live questions from speakers who have access to the Hangouts that this is being generated from. And we'll also have live questions from members of the audience here in San Francisco. But then again, we also have the questions that are coming in through the Q&A link on the site. And I'll be trying to make all of that work. So we have 5 speakers in this session in the following order. First, we'll have Ken Hayworth of the HHMI Janelia Research Campus, then Tony Zador of Cold Spring Harbor Labs, followed by Adam Marblestone of MIT and Kernel, then Ted Berger of USC, and finally Sean Mikula of the National Institute for Physiological Sciences in Japan. Okay, so I see that Ted Berger is probably getting set up over there. So we'll take about a minute or two right now, just a pause to make sure that Ted Berger is all set up. And then, oh, actually, no, it's not Ted Berger's turn yet, so no worries there. Sorry, that was my mistake. Yeah, Ken's going next. That was my mistake. I was seeing Ted getting over there, so I was assuming for some reason that would change the schedule, but we didn't. Okay, so now I'm going to introduce Ken. Our very first speaker, Dr. Ken Hayworth. Ken is a senior scientist at the Howard Hughes Medical Institute Janelia Research Campus. He's also the president and co-founder of the Brain Preservation Foundation, and he's probably one of the scientists with the longest history of carrying out explicit research with the aim of making whole brain emulation possible, and a longtime friend of mine. Ken, please feel free to begin sharing your screen to present.
Thanks, Randall. Okay, I hope people can hear me and can see my screen. Randall, can you give me a little feedback and let me know that it's working?
I can certainly hear you. Your screen hasn't popped up on the live stream yet. Probably a slight delay. Actually, I'm not seeing your screen on the Hangouts either. Are you sure you're sharing your screen?
All right, let me try that again. It says that I am presenting to everyone.
Oh, okay. That's odd because I still see your face.
Okay.
Did you select to share your whole screen, or did you select a particular window?
I selected to share the whole screen. All right. Again.
Okay, something's happening. This is good. No, it didn't.
Okay, am I back in?
I now see, let's see, I still see your face.
Share.
Now I see something different. This looks like it's your screen. Yes. Okay.
You see my slides?
Yes, I see them, and I'm going to mute myself now so that you can speak.
Great, thank you very much. Okay, so this workshop is about human mind uploading, a great topic. I probably don't have to say this, but the goal of human mind uploading is overwhelmingly difficult. In fact, it is so overwhelmingly difficult that its achievement is at least many, many decades away, as Randall's last slide was showing. And therefore, it doesn't make too much sense to talk about it, except insofar as it can motivate today's research milestones. So with that in mind, I want to start off with a speculative timeline that I think can help organize some of these research milestones. So this is going to be quite a bit different from the timeline that Randall just put up, but we can discuss that later. So here we are in 2018. If I had to speculate, I would say that the first successful human upload would not occur before the middle of this century. And it will require tens of billions of dollars of research project on the order of an Apollo moonshot. And it will probably take till late this century until the cost comes down sufficiently for quote unquote normal people to have a chance at experiencing uploading firsthand. But there are many milestones along the way to this first human upload, and these are what we should mainly be concentrating on now. That is, making sure that they occur and trying to accelerate their progress. Proof of memory decoding from a small connectome is probably the top item that that should be concentrated on, and people are working on this very seriously today. And I expect that a convincing proof of this will be demonstrated early in the next decade. Following that, imaging a whole mouse connectome. Again, people are working seriously on this, but it is really beyond the bounds of today's connectomics imaging techniques. So there's some serious developmental work that needs to go on to address this, and that's what I'll be talking about in my talk. The successful uploading of a mouse, and I'm putting that at about 20 years from now. Because this is a lot of basic neuroscience knowledge will have to be gleaned in the intervening decades to make this possible. So this is really quite out there. And then imaging the first human connectome, I would put that out at about 30 years if I had to speculate, because it is just such a monumental task. Now, I see these as the major technical milestones leading to the first successful human upload. But there are a ton of neuroscience ones that I would love to go into detail, but I can't really in this talk. A question that should be asked of any tens of billions of dollar project should be, what good is this to humanity? And Randall gave some ideas of that, but I think one that I would like to put forward is that I think the first successful human upload will usher in an era of what I might call experimental philosophy. A revolution in our understanding of what consciousness is, what the self is, what states of mind are possible. This is what really gets me excited about mind uploading. When we have complete access to an uploaded volunteer's brain, the Neil Armstrong, if you will, of mind uploading, they will be able to explore the depths of consciousness in ways that we can't even imagine. These explorations of the uploaded mind will give us, all of humanity, insight to the next stage of human evolution. So I think crystallizing the impact of that first human upload, even if it is a tens of billions of dollar project, is probably a good milestone to motivate the earlier ones that we really should be focusing on today. Now, given the current pace of neuroscience and technology, I don't think this timeline is completely unreasonable. It may be overly optimistic by a factor of 2 or 3, So why are so many neuroscientists reluctant to embrace that mind uploading is clearly the end goal of a successful neuroscience? Why are so few pursuing these early milestones? Well, here's my speculative take on this question. Joe, neuroscientist here, we'll assume he's a middle-aged or elderly distinguished neuroscientist, understands enough neuroscience to understand how monumental the task is. Mind uploading is just incredibly monumental task. And he understands that he will be long dead before mind uploading is achieved. And we all have a tendency to say things that things are impossible if they are only going to happen after we are dead. But by its very nature, mind uploading has the potential to change this. If one could come up with a method of preserving the brain of terminally ill patients today for long-term storage, then anyone alive today could be expected to bridge that gap, a gap that might be 100 years long or more. Thankfully, the technology to bridge this gap is available today in the form of brain preservation by aldehyde-stabilized cryopreservation. The ASC technique was invented by Robert McIntyre and Greg Fay and published in this 2015 article. The ASC procedure is quite straightforward. The animal's vasculature is first perfused with the deadly chemical fixative glutaraldehyde for about 30 minutes. Glutaraldehyde almost instantly stops the metabolic activity and begins cross-linking all of the cellular proteins into a sturdy mesh. After 30 minutes, they start to slowly introduce cryoprotectant into the fixative perfusate and ramp up its concentration to about 65% over the course of the next 4 hours. All of this is done at room temperature, allowing for uniform cryoprotectant concentration in all cells. This uniform high concentration of cryoprotectant allows the brain to be stored solid at extremely low temperatures, minus 135 degrees Celsius, without ice crystal formation. Now, fixation by glutaraldehyde is known to preserve the patterns of synaptic connections among neurons and to preserve the ultrastructural details of synapses. In fact, it has been the gold standard for electron microscopy for decades. And it is important to understand that glutaraldehyde is also known to preserve the primary structure and relative locations of most proteins, as papers like this and this one describe. The key point is that glutaraldehyde fixation is simply the best method known to science to preserve both the structural and molecular information across an entire brain. As a result of this cross-linking, a glutaraldehyde-fixed brain is immune to biological decay processes and will remain stable for months. But eventually, diffusion would result in the slow dislocation of biomolecules, for example, membrane lipids, that were not cross-linked. But even this slow diffusion is halted in an ASC-preserved brain stored solid at minus 135 degrees Celsius. Time has essentially stopped for such a brain. Here is a video of a rabbit brain preserved by ASC being lowered into a minus 135 degree Celsius cold storage chamber. And here is that same brain removed from storage the next day. Notice that it is completely solid. When this brain was rewarmed, we sliced it up and prepared the tissue for 3D electron microscopy. The ultrastructural details of the brain were as well preserved as those in control brains that had undergone only the normal glutaraldehyde perfusion fixation. Here is a 3D electron microscopy video of a small piece of that ASC preserved rabbit brain. This dataset was acquired by me using focused ion beam scanning electron microscopy. There is no ice damage seen, no cracks. The ultrastructural details of synapses are clearly visible, and the connectome is clearly traceable. Now, the key points to note: ASC preserved the structural connectome across the entire rabbit brain, including ultrastructural details of synapses that should make the determination of their functional strength possible. ASC preservation is compatible with the later tissue processing steps needed to map the structural connectome. And this, to me, means that ASC should be compatible with future mind uploading. This same ASC technique has now been applied to whole pig brains. These are pictures of an ASC preserved pig brain that was rewarmed after being stored at minus 135 degrees Celsius. Again, there was no macroscopic damage seen, no ice crystal damage, and no cracking. And the ultrastructure across the entire brain looks well preserved. This technique could just as easily have been performed on a human patient, and the result would have been a preservation of the structural and molecular information contained in that person's brain, and compatible with storage that can get that information to the distant future. So the implication of this that I want to stress is that ASC preservation of human patients will almost certainly support the possibility of future mind uploading. ASC is simply glutaraldehyde perfusion of the brain combined with indefinitely long-term cryostorage. ASC has been proven to preserve the structural connectome. ASC should preserve the locations and primary structures of key neuronal proteins like ion channels. ASC storage is resilient to short-term rewarming because glutaraldehyde fixed brains are stable for months at room temperature. And when rewarmed, ASC preserved brains are in an optimal state to undergo the types of tissue preparation steps likely necessary for mind uploading. They can be reperfused with lipid fixatives, heavy metal stains, and potentially more targeted stains designed to highlight particular classes of proteins. And they can be dehydrated and plastic embedded, allowing for precision sectioning and volume electron microscopy. The implications, as I see it for today's discussion, is that mind uploading is very difficult, and may not be available till next century. Because of this lack of near-term benefits, it is difficult to attract the interest and funding necessary to reach even the near-term milestones. In contrast, brain preservation is easy and could be made available in every hospital today, giving hopes to millions of terminal patients. I believe that once people begin to realize this, it will create a virtuous cycle. More people interested in personally preserving their brains will lead to more funding of research to determine what would be required for mind uploading. This will in turn lead to better evidence that mind uploading will work, which will close the circle, creating more people interested in preserving their brains. So with that preamble, let me spend the rest of my talk focusing on these two milestones: imaging mouse and human connectomes. This is a figure from Sean McCullough's recent paper, which highlights just how daunting a task we are facing. The field of connectomics is currently struggling to image a single cubic millimeter of cortex as part of the well-funded IARPA Microns project. It's a $100 million project, I believe, funding 3 different groups, and they are all struggling to image the entire connectome of 1 cubic millimeter. To put this in perspective, a whole mouse brain is 450 cubic millimeters. And a human brain is over 1 million cubic millimeters in volume. So it's a daunting task. Let's take a look at today's main connectomics imaging technologies and see if any of them are up to that task. They aren't, by the way, but I will go through them anyway. Each of these technologies has proven its ability to map the connectomes of small pieces of brain tissue. Only two of these, SEM and Atom SEM, are currently in the running for the IARPA 1 cubic millimeter challenge because they are the only ones currently compatible with high current electron imaging. Serial section transmission electron microscopy allows fast imaging because the electron beam is spread across a tens of microns wide field of view, allowing an entire image to be taken simultaneously. This has the advantage of having orders of magnitude more electron current available for imaging than, for example, a single beam scanning electron microscope. But of course, the disadvantage is that the sections must be incredibly thin, and such sections are inherently fragile and difficult to collect, even when using automatic tape collection, for example. Atom SEM collects sections on a solid tape. It allows fast imaging because it is compatible with the Zeiss multisim microscope, which is a scanning electron microscope which has up to 91 electron beams imaging the sample simultaneously. And really, this multi-beam scanning electron microscope, which I think Sean McCullough will talk about much more later, is the thing that I think will break open the mapping of connectomes. The current electron microscopes, the current Zeiss multisims have 91 beams, so that's about 91 times faster than a single electron scanning electron microscope. But Zeiss is working on hundreds of beams in a single microscope. So I think this is a rapidly advancing imaging technique. So Atom SEM, it's important to understand, is compatible with the Zeiss multisim because it collects many sections, and these are spread across a wide wafer, and so you're able to image all of those very efficiently. Again, though, it has to collect those incredibly thin sections, and they're inherently fragile and difficult to collect. Now, the Z resolution of both of these techniques is determined by the section thickness. And this represents a fundamental Z resolution versus reliability trade-off. At the resolutions needed for tracing neural circuits, which is less than 40 nanometers, probably considerably less than 40 nanometers, this means that these techniques must collect hundreds of thousands, and in a human case, millions of fragile ultra-thin sections. For this reason, I don't believe either of these techniques can credibly be scaled up to whole mouse brain level, let alone human brain level. This brings us to the two block face imaging techniques. S-beam, S-B-E-M, uses a diamond knife to scrape an ultra-thin layer off the surface of a sample block prior to each scanning electron microscope image being taken. That's how it gets its 3D volumes. And FibSim essentially works the same way, except the diamond knife is replaced with a focused ion beam of gallium ions. Both of these techniques avoid the Z resolution versus reliability trade-off because they do not require handling fragile sections. In principle, either might be able to be scaled up all the way, but in practice, it is still difficult to see how either could be scaled up to whole mouse brain level, let alone whole human brain level. Personally, I think the focused ion beam approach has the most potential to be modified into something that could be scaled up all the way, which is why I will concentrate on it for the rest of the talk. Now, because it is based on ion milling, which means it has essentially no moving parts, FibSim, focused ion beam scanning electron microscopy, can easily achieve isotropic resolutions below 10 nanometers, as this volume through an optic lobe of the fruit fly shows. We have even, at Janelia Research Campus, we have even acquired FibSim datasets with 4 by 4 by 4 nanometer resolution. So no matter what resolution is eventually required for mined uploading, it seems that ion milling approaches will be able to meet it. A significant limitation, however, is that the width of the sample in the direction of the focused ion beam is generally limited to less than 50 microns. This is because the Fib beam is hitting the sample surface at a glancing angle, which is necessary for the smooth polishing to occur. We got around this limitation by cutting larger samples into thick slabs using a heated and lubricated diamond knife. This is a video of this hot knife cutting procedure. The hot knife sections are typically 20 microns thick, which means they are incredibly reliable to cut, and there are comparatively few sections to handle compared with ultra-thin sectioning. And this is an overview of the procedure. A large block of tissue is cut into 20 micron thick sections. These are individually mounted on tabs so that they can be focused ion beam imaged with their thinnest dimension oriented in the direction of the ion beam. And this is the what an image of one of these tabs looks like looking down on the surface of it. And you can see that the hot knife cut sections, edges, are extremely smooth. I've been cutting whole fly brains using this technique. And the individual sections are then imaged across multiple FibSim machines in parallel. And the individual imaged sections are then computationally flattened and stitched back together to create a full volume. This is a zoom-in movie on one such stitch between two separately imaged hot knife thick sections. You can see that there is a boundary right down the middle where there's a stitch line. You can see that that the neuronal processes are easily traced across this cut. And here is a side view of 7 such sections spanning the entire central complex of a fly brain, all stitched together. And here's just an example of tracings performed by Varen Jain's group at Google, automated tracing of this data set. Now, but the Zeiss multisim has a field of view of over 200 microns in diameter. So FibSim cannot even clear off a single multisim field of view. And FibSim is also many orders of magnitude slower than the multisim imaging. Thus, FibSim seems fundamentally incompatible with multisim imaging. So that limits us for the 4 existing techniques. So I believe that we need a new technique for connectomics imaging. What I believe what is needed is a top-down broad ion milling approach that can mill a few nanometers thick layer off of a hot knife cut thick section. So, for example, you would have you would cut the whole, in this case, human brain coronally at 20 micron thick slices. Those 20 micron thick slices would each be put onto a silicon wafer. A broad ion milling beam would be used to mill just 10 nanometers off of its surface. Then it would be transferred into a Zeiss multisim. The surface would be imaged in its entirety. And then it would be transferred back to perform more broad ion milling. And this cycle would be repeated up to 2,000 times across this 20 microns of depth in order to create a 3D image of this 20 micron thick hot knife section at 10 nanometer isotropic resolution. To image a whole human brain this way, it would first be hot knife sectioned at 20 microns thickness into about 5,000 slabs, each mounted on its own wafer. 5,000 is a large number, but not incredibly large. And these wafers would be shuttled between multisim machines that are and ion milling machines in industrial scale robotic imaging machines. It would require several thousand multisims operating for several years straight to image an entire human brain this way. It only require one operating for about a year to image an entire mouse brain. So the entire operation, in this case for a human brain, a modern semiconductor fabrication plant, complete with automatic shuttling of wafers. This is, by the way, where I get the $10 billion figure estimate for the first human upload. Now, I've been nurturing this idea of broad ion milling for several years, but I could not figure out a way to get the broad ion milling to work. But over the past year, I have finally built a small test rig that I think is finally getting some promising results, and I'm going to show those first here. This is the first proof of principle test of this broad area ion milling SEM technique. It's on a single beam system, and it's early days on this, but I think this is a good proof of principle. This is a volume acquired at less than 10 nanometer isotropic resolution, spanning a series of 3 1 micron thick sections. So in conclusion, I think there is a path to whole brain, whole mouse brain and whole human brain connectome mapping that involves hot knife thick sectioning with ion milling and with the Zeiss multisem. I would like to think that such a technology might actually be the one that enables the first human upload, but we are talking quite a few decades before that would occur. And I want to stress again here that every step along this path to the first human upload is a step closer to proving that persons preserved by aldehyde stabilized cryopreservation can be revived in the future. Thank you.
Thank you, Ken. Just 1 second until the live stream gets there. Okay. We've reached the end. Thanks. And I'm going to give you some applause right now.
Okay. Thank you.
It's hard for you to hear the applause from here, perhaps, but there we go. Now, this is going to be a little weird locally, because to hear Ken's answer, we have to have that on. Let me just turn it off for a moment. This is going to be a little weird locally. Okay, so because I'm going to moderate the questions, and you know, online, all of that seems totally normal. Here, the live stream comes in 10 seconds later. So after I ask a question or present a question to Ken, then I'll turn on the volume again and we'll listen to his answer. I wanted to start right away with a question that came in from the online viewers. In this case, it's Michael Andrag, actually. He's asking the question, with the FibSem approach, how much damage to the sample is there from ion scattering?
Yeah, so in terms of damage, I'm not exactly sure what you mean by damage. It destroys the entire sample while it's being imaged. So there is no going back and imaging that. I'm not exactly sure what you mean by damage. It destroys the entire sample.
I'll get you the next question in a sec.
So there is no going back and imaging. I'm not exactly sure what you mean by.
So, is there a question from this audience, by the way? Very good question. What's your name, by the way? Daniel. Daniel asks, would it be possible to collect molecular information from that which is being scattered during the process so that you could use that to further identify what you're scanning at that moment?
Yeah, that's a great question. So there is a technique called secondary ion mass spectroscopy that does exactly that. It collects the great question. So there is a technique called secondary ion mass spectroscopy that does exactly that. It collects the. So I think the answer is, in principle, yes, but how I like to think of this is that the structural connectome is probably sufficient for mind uploading, and that could lead into a very, very long discussion. But in essence, the idea is that the structural connectome has so many, is so high resolution, has so many correlated features that you should be able to infer molecular information. Now, let's assume that that's not the case, though. Let's assume that we go through and do the initial experiments and show that we really do need additional molecular information. That additional molecular information is probably going to be of very specific types. It's going to be of we need to know the density of ion channels on these particular neurons. That is almost certainly what it is going to come down to. Give me the density of ion channels on particular classes of neurons. And that's the additional information. If that is the case, then I think the more proper way of doing this would be to design some type of tags, molecular tags, using immunostaining or something to that effect, that would stain the original brain. And this is why I think aldehyde stabilized cryopreservation is probably a very good technique to allow this, because you preserve the brain with glutaraldehyde, but then you stop it at that stage before going through any of the other staining steps. And so in the future, 100 years from now, they can rewarm that brain and put through whatever staining they need to highlight those particular proteins, as well as the membranes, etc. And so I think that is really the way that molecular information will be brought out. It will be highlighted with particular tags that are visible under the electron microscope, and during the scanning for the structural connectome, those additional tags will be able to be read. That gives these very specific ion channels that are necessary the ability to be to see what their densities are in particular places in the brain.
Necessary the ability to be to see what their densities are in particular places in the brain.
Thank you. I've got another question. This one is from the online audience. I've got to turn this off for a second here. Okay. This is a question by Odiseo, and he's asking, are you joining efforts with Max Moore from Alcor?
So I don't know what's happening with Alcor. I haven't talked to them in a while. Here is my take. I'm not joining efforts with anybody. Actually, I'm trying to get the best preservation possible in hospitals. Whoever does that is fantastic. What I do require for my stamp of approval, if you will, is that the whatever technique is used needs to be demonstrated and published in the literature to preserve the connectome of the brain. And whatever technique is applied to human beings needs to be needs to have a whole list of quality control checks built in. And I don't see that happening at Alcor. Maybe they could make that happen, but I don't see that happening at Alcor, and I don't see that happening anywhere else. And so what I'm really looking for is I'm looking for the larger mainstream scientific and medical community to buy into this, to say, yes, this makes sense. We need to do it right, and we need to integrate it into mainstream medicine. That's what I'm really. That's the kind of introduction that I am looking for before this is ever applied to human beings. So aldehyde stabilized cryopreservation has not been applied to any human beings. I wouldn't recommend it being applied to any human beings before those basic medical procedures have been published and the medical community has had a chance to review them for the quality checks that are necessary.
Procedures have been published, and the medical community has had a chance to review them for the quality checks that are necessary.
Thank you, Ken. We have time for one more question for you, and before I check for another one from the online community, I'd like to, Ted Berger has a question.
So I'm not sure if I missed something, but what I saw was evidence for structure, but not connectivity. That's very different.
Okay, Ted Berger says he wasn't sure if he missed something in your presentation, but what he saw was evidence for structure, but not connectivity.
So, I've looked at these 3D electron microscopy volumes, and I've shown them to many of my colleagues that trace neural circuits in normally prepared tissue, and all of them have said that the connectivity is being preserved in this aldehyde stabilized cryopreservation, these samples.
Thank you, Ken. I think that Ted Berger probably still had follow-up questions, but because we have to move on to the next speaker, we can take that to the lunch break or whatever.
Okay.
I thank you very much for your contribution once again. Thanks also for stopping the screen share at this time so that we can switch to our next speaker, whom I will introduce right now. Our second speaker today is Professor Tony Zador. Tony is the Alla Davis Harris Professor of Neuroscience at Cold Spring Harbor Laboratory, unless that's changed recently. He's also a co-founder of the prominent Cosine Conference. In the context of today's workshop, Tony's extremely interesting contribution is an altogether different approach to connectomics using molecular biology to map the brain. Tony, please feel free to begin sharing your screen to present.
Okay, let's see how this works.
Biology to map the brain. Tony, please feel free to begin sharing your screen to present.
Okay, let's see how this works. Am I sharing? Is it working? Can I get some feedback?
Looks good.
Oh, good. All right, well, let's start. All right,
yes,
so
it seems to work. Good.
So thanks for the introduction. Thanks for that first talk. It was fascinating. So I'm going to be talking indeed about a different approach to uploading the mind eventually to connectomics based on molecular approaches, and it's summarized here in the first slide. Basically, our idea is that we can use molecular biology to uniquely label all the cells in the brain, then grind up the brain in one way or another, and use high-throughput DNA sequencing to read out the projections and connections of the brain. I should say as a caveat right now that we haven't achieved that goal yet. It has the potential plus side of much higher throughput, but there's a lot of technology development that needs to happen before that works. We're getting very close to making it work in the mouse, and there are some pretty more fundamental, but perhaps not insurmountable challenges to getting it to work in humans. All right. So I added one slide sort of in the context of this talk, and just wanted to give some thoughts about the problem of uploading a brain. I think the bad news is that it's going to be very, very hard to copy every neuron, every synapse, potentially every protein. And even if we did that, even if we were able to measure all of this, it would be very challenging to know what to do with that. Would we make a huge simulation? If we made a huge simulation, we would probably have to have very strong opinions about which of those components was important in order to sort of make up for the properties we didn't measure. I think the good news is that maybe we don't need to do it exactly that way. So I'll point out that the brain that each of us walks around with today is not the same as the one we were born with. It's not the one we had 25 or even 5 or even 1 year ago. And yet we have this illusion of identity continuity. We feel like we're at some level a continuous extension of the person we were when we were born, and 5 years ago, and certainly yesterday. And so I think that suggests that there's a path to uploading a brain in which we introduce some kind of a system that interacts with and mirrors our brains during life. And such a system would have the potential to sort of represent the substrate for a permanent uploading of our minds into potentially a very different kind of a system. But I'll say that I think we have little hope of uploading a brain if we don't understand many of the fundamental principles of brain function that we absolutely do not yet understand. I would say that we know a great deal about individual neurons and synapses, and we have very little understanding of how they work together to produce thoughts and feelings and identity and memories and consciousness. All right. So what we need to do is we need to understand simple circuits. And I like to sort of put a historical perspective on this and just remind people that the first person, I think, to really study circuits in the way that many of us are trying to do these days and tease apart the circuit was Descartes, who proposed a circuit by which a person withdraws his foot from a fire, a reflex, if you like. And the mechanism that he proposed was that the flames of the fire displace the skin, which pulls on a tiny thread. The thread opens a pore in the ventricle, and then the animal spirits flow from the ventricle, which is labeled F on the diagram there, through a hollow tube, and the animal spirits inflate the muscle of the leg, causing withdrawal. Now, many of you may recognize that our modern interpretation of what goes on differs in some ways. We no longer believe in the precise location of the source of the animal spirits F. But nevertheless, I think Descartes did make an important move forward here by actually writing down a potential circuit that we could, in principle, copy and use to build a brain. And I think that's the goal of many of us working on this. So why is circuitry important? Again, I don't need to belabor that point for this group. I'll just say that we want to be able to reverse engineer the brain. And to do that, I think we need to not only make a lot of measurements, because I do think those measurements are important, and many of the and develop new technologies to make those measurements, but we have to really think hard about how the circuits that we get out by making those measurements give rise to the kinds of things that would ultimately be needed to build a brain. And then the other really immediate need for understanding circuits is that most neuropsychiatric disorders arise from disruptions of neural circuitry. Autism, schizophrenia, depression, addiction, bipolar disorder, they're all probably, or many of them are probably in part, in large part indeed, caused by disruptions of either structural or functional circuitry. And the incredibly limited set of tools that until very recently we've been forced to work with have really limited progress, to the point that, as many of you may know, major pharmaceutical companies have largely given up the search in their own labs for discovering new drugs. And it's because what big pharma is good at doing is making a drug, not finding targets, and we don't even understand what those targets are. And that's really emphasized by the fact that really in the last decade or two, really the last decade, the major frontier in treatments of of neuropsychiatric disorders have been in deep brain stimulation, circuit-based treatments that exploit the little that we do know about the organization of circuits. And so if we could know more about those circuits in mammals, in either humans or in models of brain function, that would be incredibly useful. All right, so why is circuitry hard? And that's shown here. The problem is simply that the axons are all tangled together. At least that's not simply, but that's really one of the big challenges. You see a bunch of neurons here, a bunch of wires. I'm not sure if you can see my cursor, but all those wires are somehow related to neurons depicted or shown here, and also neurons not on the screen. And we really need to be able to track them. Of course, with serial electron microscopy, that's increasingly possible, and yet that remains very slow. And the challenges are not just in the data collection, but in the data analysis. And although that technology is improving rapidly, I'm going to propose that there's a shortcut that complements the EM approach. Okay, so our core idea is to recast connectomics as a problem of high-throughput DNA sequencing by labeling each neuron with a unique sequence, a DNA barcode. And the idea is that way we don't have to trace neurons, merely read the identity of a neuron at its soma and at any place we wish to look, and we don't need to look at all the intervening sections. And that greatly simplifies the problem, and it allows us to exploit the tremendous improvements in DNA sequencing technology and more generally molecular biology. So why sequencing? Because sequencing is fast and cheap, and many of you may be familiar with this, with the fact that maybe aware of the fact that sequencing has gotten cheap, and yet it's still striking when you look at this graph to see just how quickly DNA sequencing technology has improved. So this is on the x-axis is time since 2001, which is when the human genome was sequenced, and on the y-axis is cost. And for this is cost on a log scale. So Moore's law on this is a straight line. Moore's law is a pretty healthy clip of improvement, and yet you see that the cost per genome has blown away Moore's law since about 2008. And so basically, any approach that you can sort of hitch on to the incredible improvements of DNA sequencing seems to me to be a win. All right, so right now what I'm going to focus on is the form of this technology that actually works really well right now in my lab today. We've set up a core facility. We offer this as a service now to collaborators, and that's projectome mapping. That's the mapping of long-range projections at high throughput at single neuron resolution. And to put this into perspective and motivate it, I'm showing here a slide from the Allen Brain Atlas. So what the Allen Institute did was a huge service to the entire mouse neuroscience community, which is they systematically went through and made injections throughout the mouse brain of a virus encoding GFP. So if they make the injection there on the right, they wait about 3 weeks or 4 weeks for the neurons that take up the virus to express it all through their axonal terminals. And then you can look throughout the brain, and anywhere you see green, you know that the neurons that you've injected send a projection. And they did this systematically, one injection per mouse, localized as far as they could to a single brain area. And then they, for each of those 1,000+ mice that they injected, they sliced the brain, took pictures, and put the pictures up online. And it's been an incredibly useful resource to all of us who work on this system. But that said, it has limitations. The challenge is that bulk injections don't distinguish single neuron projection patterns, and that's illustrated here. So On the left, you see an injection site. Those 3 circles are 3 neurons, and it could be that if you see green in the 3 target areas shown here, it would reflect the fact that neuron 1 projects exclusively to area 1, neuron 2 to area 2, and neuron 3 to area 3. Could also be, instead of a one-to-one mapping, an all-to-all projection mapping, or the brain being the brain. Most likely it's complicated, and that complexity actually is important to understanding what's going on. And that's the complexity that is missing when we use these kinds of bulk injections. And so the alternative to these bulk injections is to do single neuron fills, but that's incredibly low throughput. So here's a wonderful paper studying the projections from the cortex to the striatum in rats. One, for the purposes of this, what's important is that it's 1 neuron per rat. And this very nice paper showed the reconstructions of 25 neurons from 28 rats. This is a group that, at least at the time, had, I mean, it's a very skilled group. Since then, throughput has increased a little bit, but it's still nothing, not going to allow us to map all the neurons in a single brain at any throughput. So the inspiration for our approach actually came from a technique developed by Jeff Lichtman, Josh Sains, and published in 2007 called Brainbow. And there the idea is that to make the tracing easier, it would be convenient if the axons had different colors, if you could color code them. And there's this brilliant idea of coming up with a very clever genetic trick for endowing each neuron with a randomly selected collection of 3 colors drawn from a palette of about a dozen. And if you work out the combinatorics given by the genetics they did, that led to a theoretical diversity of about 200 colors in principle. And that would actually be pretty nice. But unfortunately, color space is limited and hard to read. So although the theoretical diversity was 200, the number of resolvable colors that you could actually resolve with real microscopes, actually it's not even an issue of the microscopes, it's the overlap of fluorophores in general really is closer to, it's less than 10 generously, it's probably closer to 5. And that's shown here, where those 3 purples, those 3 purple neurons are probably different purples, but they can't be resolved. So basically the core idea here is to replace colors with DNA sequences. And the reason is simply that there are a lot of possible DNA sequences. So we can kind of think of MapSeq, which is the name of the technique we developed, as a kind of digital rainbow where you replace colors with numbers and have each neuron express a unique sequence of DNA, a genetic barcode with a theoretical diversity that is essentially unbounded. And just to drive that home, there are 4 nucleotides, A, T, G, C. If you have a barcode, a random barcode of length 1, you have a potential diversity of 4. Of length 2, it's 16. And by the time you get up to 30, you get a potential diversity of 4 to the n. I'm sorry, of 10 to the 18, which is vastly more than the number of neurons in a mouse brain, a human brain, or for that matter, even in the human body. So a random 30mer is sufficient to uniquely label every cell in your body with very, very low chance of double labeling. More concretely, 10 to the 9 neurons are sufficient to label all 10 to the 7 neurons. 10 to the 9 barcodes are sufficient to label all 10 to the 7 neurons in a mouse cortex with pretty high, with sufficient diversity so that the chances that 2 neurons will be labeled the same by chance are pretty small. And in practice, how do we do that? What we do is we've generated a virus. Right now we're working with a particular virus called Synbis. And viruses are generated in the lab from a plasmid. And so to generate a viral library, all we do is we take the plasmid that generates the virus, and we introduce these random sequences directly into the plasma, into the plasmid backbone. So we get a plasmid library, and then those plasmids give rise to a viral library of sufficient diversity. There's this remaining challenge of getting the barcodes to the synapse, because typically most RNAs don't travel to distant axons. And here in one slide, I'm summarizing the PhD thesis of a very talented graduate student in my lab, Ian Pycon, who figured out a modification of a synaptic protein that is shown on the far right that allows that protein, which normally traffics to axon terminals, to grab onto the RNA barcode through a so-called N-lambda domain. And it grabs onto the box B component of the RNA barcode that we put in and drags it out to the synapse. So our first proof of principle of this was to the locus coeruleus. This was published, I guess, a year and a half ago now. And what we did was we studied the projections of the locus coeruleus throughout the brain. It was known already that the locus coeruleus projects throughout the cortex relatively uniformly, but it wasn't known whether it was suspected and believed, but not really known, that the neurons in the locus coeruleus just kind of bathe the cortex in noradrenaline. So the locus coeruleus is the sole source of noradrenaline to the cortex. Noradrenaline is a kind of wake-up signal. It tells the brain to go on alert. And the usual view is that there's a cortex-wide alert signal that goes off when the locus coeruleus is activated. But alternatively, one can imagine that the locus coeruleus actually can specifically activate subparts of the cortex. So for example, if you need to specifically listen for sound, you might imagine that the auditory cortex is selectively turned on. And maybe if you need to selectively be on the alert for light, it might be the visual cortex. So I'm going to skip over some of these details and just say that for the most part in these experiments, it turns out that the RNA acts like GFP. It acts like a bulk signal that bathes the brain. And the way the experiment goes is we inject in the locus coeruleus, that cross in the back there, and then cut the brain into slices. And then we extract the RNA from each of the slices and figure out which barcodes appear in each of those slices. And then we can demultiplex that, and we can get a picture that looks like this, for example. So here is the profile, the spatial profile of projections for barcode 28, which corresponds to neuron 28. And the projection strength is measured in barcode molecules, which we quantify carefully through sequencing. And the x-axis shown here is the rostrocaudal position from those slices. So it's important to recognize that with this technique, the spatial resolution is determined by the at the time of dissection. So we collapse all the barcodes that are found in whatever chunk of brain that we collect and put them and can't resolve them further. So this particular neuron you see has a very specific spatial pattern of projections. Another neuron, and that's at odds with the traditional model, another neuron has a much broader one, barcode 79, and there are even more complicated ones like bimodal projections. And in all, we were able to get 1,000 single neurons and their projections from just a handful of animals in just a couple weeks. So this is orders of magnitude more throughput for this system than has ever been. Well, since there were I think there were no locus coeruleus single neuron reconstructions, so more than orders of magnitude is infinitely more. But in general, 1,000 neurons in a handful of experiments is high throughput. We've since then, just a technical note, many of you are probably worrying about double infections. What if you get two barcodes into the same neuron? And in the interest of time, I'm not going to go over why that's not bad, but it isn't bad, and you can ask me during the questions. What would be bad would be degenerate barcodes, insufficient barcode diversity that would end up with the same barcode ending up in 3 different neurons, and that would be disastrous. So we go out of our way to avoid that, and then we confirm that we avoided it. So if people have these technical questions, I invite you to ask me during the question period. I'll also say that more recently, we now have in collaboration with Tom Merzick-Flogel's lab, we did who did a bunch of single neuron reconstructions in visual cortex. We compared their reconstructions, 35 neurons in a couple of years, to some map-seq studies of those same projections, 533 neurons, in a handful of experiments. And for the purposes of this talk, what's important is that they agree to essentially the resolution that we can determine in this experiment. And that there's a whole scientific story that I don't have time to go into, but that should be coming out in Nature sometime in the next few weeks, and it's on bioRxives right now. All right, so we have a core facility. This is up and running. We're now studying the projections in different animal models, different brain areas. And we're scaling up to do the whole brain in a single experiment, and I'm just going to go through that very quickly. We tile the entire brain with, or the entire cortex with barcode. Then we take much finer slices than in the previous work, align it to the Allen Brain Reference Atlas, and then we can project these back to Allen reference coordinates. And in a single experiment, in about a week for about $10,000, we can get the projection patterns of about 100,000 individual neurons. And for each neuron, we can see the areas to which it projects. So here are the strongest projections of 2 of these 100,000 neurons, just randomly selected neurons, to give you a feel for what kind of data that we have. So this is so much data, and of a different form, that although we've had these data now for almost a year and a half, we're still figuring out how to make sense of them and what to say about them. But we're writing this up right now. Nevertheless, none of this gets us to the connectome. So far, I've only told you about the projectome. And so I want to, in the last couple minutes, talk about that. So the limitations of map-seq are that, first, it has crude spatial resolution, as I mentioned. It's limited by dissection. There's no notion of morphology or cell type. And I should say that I'm not getting any feedback on the screen right now. So I think I'm 25 minutes in, but if the MC wants to communicate with me, I'm looking at my cell phone. You have my cell phone number, and you can let me know how I'm doing on time or anything else. And if you want to communicate with me, I'm looking at my cell phone. You have my cell phone
Tony?
number. Yeah.
Yeah, you're fine. You're fine, actually.
Okay. If you want a side channel, I'm staring at my phone, so you can text me or whatever you want. Okay. In fact, that's reassuring to hear that you hear me, because for all I know, I've spent the last 25 minutes talking to my screen. I got to say, this is a strange forum where I don't see the faces of the people in the audience. You know. Anyway, so the second issue is that there's no notion of morphology or cell type gene expression. And my own background is in computation and in physiology. So the fact that anatomy is traditionally very hard to correlate or relate to neural activity, to knowing what the neurons did when the neurons were still inside the animal's head and the animal was doing interesting things, is distressing. And of course, as I mentioned, I haven't told you how we can get from the projectome to the actual synaptic connections that were needed to figure out the connectome. And the solution to all of those is a technique that we call bar-seq, that's modified from in situ sequencing, which is a technique that was developed in George Church's lab, and which we learned from them through collaboration, and then modified and adapted to the particular goals here. Okay, so to tell you how that technology works, I have to actually take you through just a little bit of the nuts and bolts of conventional DNA sequencing. Many people who do sequencing only have a vague idea of how the sequencing actually works. This is, when I say conventional sequencing, I mean conventional short read, high throughput sequencing as done by Illumina. And so this is how it works. On the left, you have a tube full of DNA, little bits of DNA that you want sequenced. You wash it onto a so-called flow cell. Those little black bars sticking up are sequences that grab onto the purple DNA that you want sequenced. And then the next step, you amplify each one of those single pieces of DNA into a little cluster on the slide of identical bits of DNA. So that's done by bridge amplification. And then the key thing is that then the sequencing actually becomes an imaging problem. So you take a snapshot of those clusters, but before taking the snapshot, you label each cluster with the next nucleotide in its sequence. So again, I'm not sure if you can see my pointer, but for example, this one in the left, the first nucleotide in that sequence might be a C, and so it's labeled with a fluorophore that's white. This one on the top is an A, so it might be pink. Same with this one. And this one here on the right is a T. Then you take a picture of this using originally they used a spinning disk confocal. They've actually changed that technology, but originally it was with essentially a confocal microscope. Then cleave those fluorophores off and add the next nucleotide. And so to build up the sequence of DNA associated with each one of those single molecules really is a matter of taking a bunch of pictures and aligning those pictures properly. And so the number of dots per picture represents the number of molecules that you're sequencing, and the number of cycles tells you the length of the DNA that you're able to get. So in this case, C, T, T, those are the first 3 bases, and then some dots. And typically these technologies can nowadays go up to several hundred nucleotides. So the key point here is that conventional high throughput next generation sequencing involves taking a picture. So for in situ sequencing, we can do essentially the same thing, except that instead of first extracting the DNA, we first immobilize the RNA in the cell, and then using a different set of molecular biology tricks, we convert each one of those individual molecules of RNA into a ball of cDNA, complementary DNA, that corresponds to the sequence we're interested in, the RNA. And from there on in, it's essentially the same problem of imaging. So here's an example that I hope will clarify what I'm talking about. This is a picture of a bunch of neurons in culture. The bright circles are cell bodies. I'm showing you these pictures from cell culture because the pictures are prettier, but we also do this routinely now in slices. And so what you see is, here's the key for reading it out. Each neuron is full of one barcode derived, or in a couple cases, 2 barcodes, but typically 1 barcode derived from infection by our virus. And just to show you how this plays out, here are 2 neurons I'll show you the sequence of. The first base is an A, then we cleave that off, the next base is a G, and so on. And so by taking a series of pictures, we can read out the sequence of barcode in each neuron, and with different optics and modifying and optimizing a couple of things, we can also do it in axons and dendrites. So we're now applying this to study the projections in auditory cortex, and in the interest of time, I'm going to skip over most of that and just say that here's a picture of about 1,500 neurons whose sequences we've determined in a brain slice. Well, we took these neurons, we took a brain, we injected the virus in auditory cortex, then we waited 2 days, then we did in situ sequencing, bar-seq, at the injection site and looked at the long-range projections using conventional map-seq. And in that way, we were able to get the projections of cell body positions of about 1,500 neurons. And so for each one of those little dots, we can click on it, and I could tell you the strength of projections to the dozen or so areas that we've studied. And again, there's a whole story that we're working out here. We can combine this with in vivo activity. We first use 2-photon imaging to figure out the activity of a bunch of cells. And then ex vivo, we can find those same cells and then figure out their projections. We can combine this with gene expression. Let's skip over that. And then here's what I think which is what's most relevant to the focus of this meeting, which is how do we achieve synaptic resolution? And essentially, I've already told you the answer, which is that we combine the axonal and dendritic labeling with barcodes in this tangled mess that I showed you. And we can figure out what the two barcodes are, the two barcodes are of the potential processes that are the processes that are forming a potential synapse. We have to actually go one step further and figure out whether there is an actual synapse there. Currently, we're doing that by using a synaptic marker. Going forward, we have some molecular tricks for actually entangling the barcodes associated with the two processes so that a single ROLINY will form that includes the sequences of the two barcodes, but only when the two barcodes are touching at a synapse. So in summary, MapSeq and BarSeq open up the possibility of combining connectivity, gene expression, in vivo function at high throughput in a low-cost way. And I'll just summarize by saying that if the brain were so simple we could understand it, we would be so simple we couldn't. And I can't end without first thanking my collaboratome, especially Eustace, Xiaoyin, Lee, and former lab members, including Ian Pycon, and also my fundosome, especially IARPA, NIH, and the Paul Allen Family Foundation for some very early funding for this. And with that, I will close.